And here we are again. Cell biology. Cell biology. Okay, do not have a quiz for you today. Sorry, I forgot about it this morning and I got busy, so I didn't write one up. So we're going to have to go quizless today. But I have some exciting news. We have more signal transduction pathways to look at. I think I told you this before, but one time I asked the students what SMAD means, and one student said, so many asinine diagrams. Kind of true. Okay. Without any more ado, you know how I hate to do. This is the wrong one. Wait. Okay. Here we are. And arranging my real estate here. So last time I covered this part, which is actually in lesson 3 from last year. So we're a little bit ahead. Let me review this again. Let me review this because we only did it once, so you can't review it twice. So basically, this is what I used to call the big picture. How do you get from hormone hitting receptor to the cell actually dividing? And by the time we get to the exam, which is in 2 weeks, I want you to know this. This will definitely be on the exam. So the first part is from the last exam, but it still counts here. And then we'll see how this connects. So your growth factors hit receptors, which then cross-phosphorylate and make SH2 domains. That brings GRB2 and SOS up. SOS activates RAS. RAS activates RAF, MEK, MAP kinase, shoot for your kinase cascade. MAP kinase goes in and then activates some transcription factors, Jun, which then binds to FOS and becomes AP1, and ETS, and other ones too, but these are the ones we're talking about. So these transcription factors then turn on immediate early genes. Again, these are coming up within 30 minutes of stimulating with the growth factor, and you don't need any protein translation to make them come up. All the protein machinery is there ready to go when you hit with the growth factor. So these IEGs come up first. They get transcribed. Their mRNA gets transported out of the nucleus, gets translated into protein. Those proteins get transported back into the nucleus. And a lot of them, like this, are transcription factors themselves. So when they come back into the nucleus, they turn on transcription of your delayed early genes. And your delayed early genes include our cyclins, our CDKs, and most importantly, the transcription factor E2F. E2F, when it's fully active, will be the thing that actually gets us through the restriction point and into S phase of the cell cycle. And this is how that happens. So E2F gets made, and it immediately gets bound up by RB. RB is a tumor suppressor. We'll talk about them here and in the next chapter. So tumor suppressor because it only causes tumors when it's not there anymore. So as long as it's present, it blocks cell division. So RB grabs E2F and blocks it. And then you get your cyclin CDK thing happening. So cyclins come up, they bind to the CDKs, they get phosphorylated, dephosphorylated, and then they phosphorylate RB. So I said earlier that this transition here has to do with three different CDKs and 2 different cyclins. So cyclin D will activate CDKs 4 and 6, and they will kind of hypophosphorylate RB. So it kind of gets it ready, but it's not really inactivated yet. And then that will go away, and then cyclin E will come up, and it will bind to CDK2, and that then will hyperphosphorylate RB. So a lot of phosphate gets put into RB, then it changes conformation and releases E2F. E2F then is a transcription factor, and you get transcription of all the genes that the cell needs to go into DNA replication, and boom, you're going forward now. And now at this point in the cell cycle, once you've passed that restriction point, the cell's no longer responsive to extracellular signals anymore of any kind, growth factors especially, but of any kind really. And now it's on its own clock. It's got its own program to go from this point all the way through back into G1 again. Okay, so that is your cell cycle. Now, in chapter 23, when we talked about growth factor receptors, I said, you know, here is our, where is it, GRB2 site. So we're activating that whole RAS pathway. But you've got other things that are also activated at the same time or in other cells, usually at the same time, but also in other cells. PI3 kinase was one of them. If you remember this pathway, and I hope you do, PI3 kinase, when it was activated, so this is again activated by the growth factor. So the growth factor causes, you know, dimerization of the receptors, cross-phosphorylation, and that brings PI3 kinase up to the membrane, and that makes PIP3. So PIP3 then activates AKT. So one of the things that's going to do is release glucose. Now, a thing about cancer cells we'll learn later is they really thrive on glucose. It's fast energy, they burn it up quick. So that's a pathway that gets activated in cancer cells. Not important here, really. What's more important for us is AKT is blocking GSK3 beta. And GSK3 beta had been breaking down beta-catenin, but when it gets blocked, it's then around and it binds to TCF-LF in the nucleus, and it becomes a co-activator with that transcription factor. And then you get production of cyclin D. So here we are, that's our DEGs. So beta-catenin then is going to cause transcription of our DEGs, and then the cell goes into the cell cycle. So that's our connection with this pathway in cell division. You also have another pathway which activates, Rheb is another kind of RAS-like G protein, which activates TOR, which we called PDK2 at the time. So just another pH domain containing kinase, and that also can cause cyclin D to come up through different mechanisms. Now, here's something totally different, which is also really important. So what I just talked about was how the receptor pathways we've talked about in the past are linked to causing the cell to go into cell cycle. Another thing you need to know about cell cycle is if a cell is overstimulated, if it is really heavily stimulated to grow, like beta-catenin increases dramatically or E2F increases dramatically, it will go into apoptosis. And that's kind of a safety valve so that, you know, cells don't get overheated and then they become cancerous because they're overstimulated. Hang on 1 second. Gus, he's up on the water there, buddy. He has a tendency to drink too much and then throw it all up on the floor. We got to remind him not to do that. Okay, so whenever the cell is stimulated to divide, it also needs to be, it also has to activate pathways that are going to inhibit apoptosis. Apoptosis is like the next thing we're going to talk about, or 2 things from now. So it's all part, apoptosis is all part of cell division for this reason. So when AKT gets activated, it also will phosphorylate BAD, and BAD is an anti-apoptotic BCL2 protein. Don't be surprised if none of that makes any sense to you, because we haven't talked about it yet, but we will talk about it today. So this is an anti-apoptotic BCL2. So this is going to block apoptosis and allow the cell to go ahead and divide, and therefore become 2 cells. Okay. All right. So that's it for connecting signal transduction to cell division. Do we have any questions there? Any comments you people might have? I must be an amazing professor because you never have any questions. Everything's so clear, so clear. Do you know who the new dean is? I think it's Jeannie Babb. Why don't you guys all write her an email and tell her she needs to give me a raise because I'm doing such a great job here. So my students never have any questions. Never. It's all so clear. Nothing? Okay. So next thing to talk about is our old friend P53. You've probably heard about P53 before. It's really important. And it's more important than in its absence than its presence, really. I mean, it's in every single cell, and it's protecting your cells from becoming carcinogenic. And so cancer, as we'll talk about in the next chapter, is all about DNA damage. So if your DNA is all good, then cells will never become cancerous. But when you get mutations in some of these proteins, like everything along that line from growth factor to E2F, anything along there could be mutated so it's permanently active. And if that happens, then you can't shut the pathway down. The cell's no longer responsive to external signals, and it's constantly in the cell cycle. That's cancer, okay? Constantly dividing cells that are no longer responsive to the needs of the body. So P53 is there to help protect against DNA damage because that's what causes cancer. And the kind of damage it protects against is really large scale kind of changes like single strand DNA breaks, double strand DNA breaks, and some chemicals can cause this. Radiation is really common is cause of double and single strand breaks. So let's see, reading from the notes, P53 is mutated in more than 50% of all cancers. Okay, and that's not because mutating it causes cancer, it's because mutating it allows cancer. Once your cells no longer have P53, mutations that normally crop up are no longer fixed, and they continue in that cell line, and then eventually they become cancerous. And again, we call this a tumor suppressor because cancer happens in its absence, not its presence. We'll talk about this in the next chapter, but an oncogene directly causes cancer, and a tumor suppressor normally blocks cancer, but when it's absent, cancer happens. So this is a tumor suppressor because it helps to protect the cell, and when it's not there, you get a lot of mutations building up. P53 is regulated similar to beta-catenin. It's constantly made and constantly broken down, okay? Why would a cell do that? Let's open this to the crowd. Let's put this up for sale here. Stop my share. I don't know if I talked about this before, but let's put this for a serious number of bonus points. 3 bonus points, first answer. And here's the thing, you have to, where is it? Reactions, go to your reactions. Give me one of these when you know the answer, and then I'll choose you. By the way, I can't see you unless your face is up. At least I can't see all of you. Okay, ready? Question is, why would a cell, I think I did talk about this already. Why would a cell make a protein just to break it down? Why wouldn't it make it when it needs it? Why wouldn't it make P53 when it needs it? Instead, it's being made and being broken down. Why? I did talk about this once before. Well, I think Claire came up first, or maybe Alexa, but I'm going to go with Claire. Muted. You're muted. Alexa's next. You're still muted. I can't hear you.
Can you hear me now?
Yes.
Okay, sorry. Alexa came up first.
Oh, okay. We're going to let Alexa go.
Okay, thanks. Can you hear me?
Yes.
Okay. Does it have something to do with you saying it takes more energy to like make it when they just need it, as opposed to just keeping the mechanism going of just continuously making something?
You're close, but you're wrong. It takes more energy to do it this way. What does it take more of to do the other way? Not energy.
ATP?
No, that's energy.
More enzymes?
Well, I mean, it obviously takes more time, doesn't it?
Time. Who said that?
Me?
Who's me?
Alexa.
Samantha and Alexa.
Okay. Alexa, yeah, your face is covered. I said it. Why?
Sorry.
That's okay. Okay, Alexa, I did give you a hint on that by humming a famous Rolling Stone song, which I was hoping Barbara Perry would get because I know that's more her generation than mine. But okay, I'll give you 3 points anyway because that's the kind of guy I am. All right, let's go
Thank you.
back. Let's get back to work. Who chatted me? Time. Samantha, I said to put your hand up. Okay, sorry, Samantha. I'll give you 1 anyway because whatever. Time is on my side. Yes, it is. Okay, so what the hell were we talking about? All right, so the whole point is if there's a problem in the cell, you want help to come quick, right? And so if damage was causing the upregulation of P53 through a transcriptional mechanism, it would take, 30 minutes to an hour for P53 to come up. But by this mechanism, it comes up in like minutes, like 10 minutes, boom. It like, I think it doubles every 2 minutes or 5 minutes or something like that. So the fact that it's constantly being made and broken down means when you stop breaking it down, comes up really quickly. So it's a much faster mechanism, although it does waste a lot of energy. And it is broken down by ubiquitination and mechanisms protected. So let's do the actual pathway now and talk about what happens. So here's P53. So on a regular basis, when there is no damage to the DNA, P53 is constantly being ubiquitinated by MDM2. So MDM2 is ubiquitin ligase. So MDM2 ubiquitinates it, P53 runs off and gets broken down. Okay, that cycle keeps going. Now if you've got some kind of DNA damage though, oops, sorry. Hang on a second, never mind. My Zoom's being annoying, but I'm gonna ignore it. If you've got some kind of DNA damage, like if you've got a single-stranded break, that will activate ATM. Let me make sure I got that right. No, if you've got a double-stranded break, that will activate ATM, which is a checkpoint kinase. It's a kinase that's looking for damage. If you've got a single-stranded break, it'll activate ATR. So these two are out there constantly looking for breaks in the backbone of the DNA. When they find something like that, they phosphorylate checkpoint kinases, which will then phosphorylate P53. When P53 gets phosphorylated, it's protected. MDM2 can no longer break it down. Now you've got 2 different paths you can go on here, depending on how much damage there is. If there's a little bit of damage, then P53 will activate a protein called P21. P21 is a CDK inhibitor. So remember, CDKs are important for the cell cycle, keeping the cell cycle going. And P21 will stop the cell cycle at any point between the restriction point and mitosis. So when P21 comes up, boom, the cell stops. And then it also brings up, it doesn't say it here, but it also brings up DNA repair enzymes. So those repair enzymes will go out, find the damage, fix it, and then once the damage is fixed, that will turn off ATM and ATR. These get dephosphorylated, and P53 will get dephosphorylated, and then it gets broken down again. Cell goes back into the cell cycle. So that's kind of like a low amount of damage. We pause the cell for a moment, we fix the damage, and then we go back to cell cycle. If you've got a lot of damage, like imagine some stupid Irishman in the summer going out without any sunscreen on, boom, sunburn everywhere, right? And really bright red skin and cell death. So what's going on there is you get a ton of DNA damage. That UV starts to split the DNA. That activates your checkpoint kinases again, protects P53, and now P53 activates a protein called PUMA. PUMA is a pro-apoptotic BCL2. So when PUMA comes up, the cell's going to go into apoptosis and die. And that's why if you get a really bad sunburn, you get blisters, because you've got, the cells underneath are dead, and all these, you get inflammation, which is the red, and all these immune cells go in there to get rid of those dead cells and repair the damage and everything. So it's pretty traumatic to your skin. Oh, if I could go back, how old am I? I could go back 40 years right now and give myself one piece of information, it would be sunscreen. My skin right now, I've got like pre-cancerous growths all over my head right now because I was like Mr. Lay out in the sun and get a tan, which is ridiculous when you're Irish because your skin doesn't really tan anyway. So anyway, sunscreen, people. All right, so just to go over that again, normally MDM2 is phosphorylating P53 and is being broken down. When you've got DNA damage, you activate ATM or ATR or both. That activates checkpoint kinases, which phosphorylate and protect P53. If you've got a small amount of damage, you make P21, which blocks the cell cycle, and DNA repair enzymes. And that will repair the damage, and then you go back to normal. If you've got a lot of damage, you activate PUMA, which then just causes the cell to die. And again, it's better that the cell dies than it keeps dividing with damage. So it's kind of a protective mechanism. Okay, questions, comments? Okay, so what is apoptosis? That was your next question, right? So probably not, but what the hell? Okay, so here's what a normal cell looks like, a normal epithelial cell, I should say. It's very flat, right? So when apoptosis gets activated, it ends up looking like this, rounded up with these big bulbous blebs coming out of it. What do you think is causing that? Someone shout it out. Why is the cell changing shape?
Apoptosis.
Well, yeah. But what's happening in apoptosis that's causing the cell shape change? What holds cell shape?
Cytoskeleton.
Cytoskeleton. So when apoptosis gets activated, the cytoskeleton gets broken down and the cell just falls apart. So these blebs are because you no longer have that cortex holding the plasma membrane together, and it's just blowing up like a balloon. And this whole cell will fall apart into individual chunks, and immune cells will come in there and get rid of it. When does this apoptosis happen? Well, one is what we just talked about, when you've got a lot of DNA damage, and that's kind of a that's kind of because of damage or whatever. There are also normal periods in development and in your life when apoptosis just happens, like especially in development. So, the developmental program up to a certain point, like once it gets to a certain point, they call that the phylotypic point, when all embryos look the same. So you've seen this embryo with a funny looking head, and then the little arm buds coming out and the tail screwing around. Everything in the phylum of Chordata looks like that. And from there, it all changes. And that's because it's so complicated getting to that point. If you have a change in the program, it will never develop, right? So you have to have everything working exactly perfectly to get to that point. And then changes can happen to the program after that. That's kind of like how new species evolve, because after that one point, things can change and you get a different shape. There's a new discipline called EVO-DEVO, evolution and development together, and how does development result in evolution, which is really interesting stuff. Um, but anyway, what the hell was I talking about? I was talking about so in development, you've probably seen when your hand first develops, you've got skin between the fingers, right? You've got webbed fingers, and those cells die back and make your digits individual, right? That's apoptosis, the loss of those cells. And that developmental program can change. If you look at my golden retriever here laying at my feet, he's got webbed feet, right? Not all dogs do, but golden retrievers do, and it makes him a really good swimmer. So that program can change pretty easily. Also, the tail, right? Most of us are not born with the tail. Some people are, some people have a short tail, but most of us, that tail dies back during development. And they call it the post-anal tail. And that's apoptosis as well. Also, if a cell is infected, an immune cell may come up and kill the cell. If it can't kill the virus or bacteria or whatever, it'll just kill the cell, and that's part of the process of getting rid of dangerous stuff in your body. What else? Involution is another one of my favorite terms. So or at least some of you know already, when a woman gets pregnant, towards the end of pregnancy, her breasts get really large, and after birth they get even bigger, and that's because they have to be ready to go. After she's weaned the children, they get smaller again, and that's involution. And again, apoptosis is killing off the cells that are not needed anymore. Because remember, all those cells are costing the organism energy. So once you're no longer feeding children, there's no point in having them there. So they die back, and then they can come back again if you have another child. Okay, so apoptosis in normal cells. So there's a 4-step process that you see in all cells when they apoptose. Blebs. I love the word bleb. Okay, so first thing that happens is the DNA condenses and the cell rounds up. So we're going from that flat cell into a round cell. Same thing we see in mitosis, but for a different reason. Blebs start to form in the plasma membrane, and that's because that actin cortex is being degraded. Especially actin gets hit early and fast. The DNA starts to fragment into 200 base pair pieces. So if you're in developmental biology, this week we talked about DNA packaging, how you've got a nucleosome, which is 2, 4, 6, 8 histones with 200 base pairs wrapped around it. So we've got a DNA that gets activated here and clips it at either end of the nucleosome. So all the DNA is in 200 base pair pieces. And the organelles start to fragment. So another thing that gets attacked early on is your mitochondria. They break down that outer membrane and then the inner membrane, and then there's no more ion gradients, and the mitochondria is shut down. And then the whole thing just falls apart into these apoptotic bodies, just chunks of cell, basically, with chunks of organelle inside them. And then Mr. Phagocyte comes along and eats up those phagocytotic, those apoptotic bodies, and takes care of the debris. Okay, so what's important here? Cell rounds up from the cytoskeleton being degraded. The DNA fragments into small pieces. The organelles fragment; they fall apart as well, and then the cell collapses into apoptotic bodies. Let's take a look at this animation. Necrosis versus apoptosis. Hello everyone. This tutorial will explain to you the main difference between apoptosis and necrosis. Introduction. All cells in a multicellular organism undergo growth and death. Cellular death is essential for an organism to grow and survive. Human body is made up of innumerable types of cells in various parts of the body. There are mainly 2 types in which a cell undergoes death, either through exposure to harmful environment, injury, or through a plant-regulated process of disintegration. Cell death occurs via 2 processes: primary necrosis and apoptosis. Apoptosis. Damn Englishman.
Necrosis is a process of cellular death, which occurs when cells are exposed to extreme conditions, which are very different from normal living conditions. This leads to damage to the internal cellular environment and rapid cell and tissue damage. So the basic inability of the cell to maintain balance when exposed to rough conditions leads to complete discord in the cell's functioning and eventually its death. What is apoptosis? Apoptosis is a pre-planned regular process of cell death occurring in the body, where the cell itself actively takes part in its own death. It occurs as part of normal cellular multiplication and turnover. So cells die naturally to maintain a balance of the cellular multiplication. Apoptosis is required for smooth functioning of the body. Most cells in the body have a shelf life. For example, red blood cells live for 120 days. At the end, they are destroyed in the body. This is a method of apoptosis. Difference in cause and presentation. Necrosis is a pathological process which is detrimental to the body. It occurs when the cells are exposed to toxins or on exposure to extreme conditions, which could be anything from increased temperature, decreased oxygen levels, etc. These conditions lead to damage to the wall or membrane of the cell. An inability to maintain normal functioning in these conditions leads to an imbalance in the internal environment of the cell. This leads to inflammation and tissue damage, which leads to accumulation of cellular debris. Apoptosis is a normal, natural process which is necessary for the body, as it maintains a balance in the number of cells in the body. If the cells do not die on time, it can lead to a tumor or cancer formation, which is an accumulation of unwanted cells. If cells die too fast, it can lead to conditions such as AIDS, heart disease, and liver disease. So if apoptosis does not take place as it should, there will be signs of disease. Difference in structural changes. In necrosis, there is a loss of integrity of the wall of the cell, which leads to swelling of the cell's contents, as well as disintegration of small bodies of the cell. In apoptosis, there is no disintegration of the cell membrane or wall. Rather, there is a shrinkage of cellular contents along with clumping of the membrane. This leads to formation of condensed bodies known as apoptotic bodies. These bodies are recognized by the immune system of the body and destroyed efficiently by the immune cells. Thus, it is a well-coordinated process due to which there is no accompanying inflammatory reaction. Difference in biochemical reactions. Necrosis is a passive process which does not require energy and can occur at any point of time. This is a random event which is unregulated. Apoptosis is an active process which requires energy and occurs in an organized manner. Many enzymes and agents are required to carry out various steps of this process. Summary. Apoptosis and necrosis are variants of cell death that vary greatly in all ways except the end result. Necrosis is a type of cell death wherein the cell dies an untimely death due to uncontrolled external factors, and apoptosis is a predefined cell suicide wherein the cell actively destroys itself to maintain smooth functioning of the body. Apoptosis is a natural physiological process which is required by the body, while necrosis is a pathological process which stems from inflammation and tissue damage.
Okay. So, what do I want to say? Oh, one of the things I liked about that was you see that layer of cells and the upper cell dies and it's replaced from below. That's kind of showing you what the intestines are like, right? In the intestines, you've got these crypts, and at the bottom of the crypts, you've got stem cells that constantly divide, and as they divide, they differentiate going up the crypt till they hit the surface of the colon, and then when they hit the surface, they go into apoptosis and die, and then they get replaced from below. So your colon is constantly being replaced by stem cells and apoptosis. So that's kind of what he was showing there. Okay, so that's all well and good, but let's see how this actually happens, shall we? Okay, so my figures kind of stink at this point. Let me go to the notes and then we'll come back to the figures. So the notes are kind of setting up how this process, the proteins involved in this process. Oosh, go away, you. Okay, so all these events that you see in apoptosis involve enzymes called caspases. These are called caspases because they have cysteine in their active site, and they cut proteins at aspartate. So cytoskeletal proteins are, aspartate is a negatively charged amino acid. So you see it a lot in cytosolic proteins, especially cytoskeleton. So when these things become active, they're going to go out and break down everything that's got aspartate in it, which is the cytoskeleton. So they go out and break down the cytoskeleton. They also activate a DNase that's going to break down the DNA. And these caspases are in all your cells right now. Every one of your cells is filled with these enzymes on a suicide mission. I keep thinking like a really cool, don't report me for this. Don't tell the FBI I'm saying this. A really cool chemical warfare agent would be one that would activate your caspases. So you'd be standing there, you wouldn't smell anything, but suddenly the chemical agent gets into your body and all your cells turn into jello all at once and you fall down and you're just a skeleton with jello dripping off of you. Isn't that cool? Don't tell the FBI. Okay, so there's two different kinds of caspases. There are, and you get kind of like a cascade activation of caspases. So you've got initiator caspases that get activated by some mechanism, and then those initiator caspases go out and activate the executioner caspases. Our most important executioner caspase is caspase 3. This one, you'll see all our pathways lead to caspase 3, and it's the one that does most of the work of breaking down the cell. In terms of initiator caspases, we've got two different pathways that lead to the breakdown of the cell. One goes through caspase 8 and the other goes through caspase 9. So another thing I should have here is these, oh, there it is right there. These caspases are in your cell as pro-caspases, which means like there's like a domain that's blocking the active site. So there's a piece of the protein is blocking the active site. So proteolytic cleavage then pulls that domain off and they become active. So the activation of these caspases all involves a little bit of protease activity. And then once one is activated, it activates another one and that activates another one. So it becomes a chain reaction of activating them. So you get this slow start and then it builds until the whole cell is filled with caspases and the whole thing just breaks down. So we've got a couple different pathways. I'm not sure if these numbers are right. I think I added some slides, so these numbers are not right. What is this? This is slide 48.
I think it's 48 through 55. Oh, sorry, I went through and tried to fix them.
Okay, I think I added 4 slides somewhere, so it's 48 to 51. I'll update this one. Thanks, Carrie. So, okay. Let's go to new share. Okay, so this is the extrinsic pathway, which means that another cell comes and tells the cell to die. So this is what I was alluding to when I talked about juxtacrine hormone interactions. That's when one cell touches another cell, and that causes activation of signal transduction. So here you've got a death receptor on all cells. This death receptor is called FAS, F-A-S. And then you've got on cytotoxic T cells, FAS ligand. So the cytotoxic T cell may be attracted to the cell because of some toxin it's putting off or some signal that's telling the T cell that there's something wrong with the cell, we got to get rid of it. So the T cell senses that, it comes over and it touches its FAS ligand to FAS, and that activates accumulation here of a number of proteins. One is called FADD, FAS associated death domain. So FADD gets attracted to FAS, and then that brings up procaspase 8. Procaspase 8 then gets activated to become caspase 8. Okay, now it's caspase 8, and that activates caspase 3, and that causes apoptosis. Okay, so you've got FAS in the membrane, which is the receptor. FAS ligand touches it, FADD comes up, procaspase 8 comes out and it gets activated to caspase 8. That activates caspase C, caspase 3. All right, so that's the extrinsic pathway. And the intrinsic pathway is quite a bit more complicated. Let me bring up my notes again because I don't have a good slide on this. So this is where we get into BCL2s that I talked about earlier today. So BCL2s are a set of proteins. The first one found was BCL2. So the whole class of proteins is called BCL2 because that was the first one. And you've got two different types of BCL2 proteins. One are pro-apoptotic and the other are anti-apoptotic. So in all of your cells right now, you've got a lot of anti-apoptotic BCL2s, or else your cells would all go into apoptosis. So BCL2s are in the membrane of the mitochondrion, and they're keeping cytochrome C inside the mitochondria. So cytochrome C is a member of the electron transport chain, and it normally is in the inner membrane or associated with the inner membrane of the mitochondria. But if it gets released from the mitochondria, it will cause apoptosis. So you've got these anti-apoptotic BCL2s, which are BCL2 and also BCLX. These are inhibiting release of cytochrome C from the mitochondria. And then you've got your pro-apoptotic BCL2s, and there are a lot of these. So we talked about PUMA earlier. That came up when P53 increased, right? It caused PUMA to come up. Then you've got NOXA, BID, BAXBAD. They all have these BIP types of names for some reason. BAD is another one we've seen already, right? BAD is phosphorylated by AKT and inhibited by AKT. So BAD will cause apoptosis unless AKT phosphorylates it. So that's what we talked about earlier today. So all of these things are pro-apoptotic BCL2s. And the way you want to think about it is you've got your anti-apoptotic BCL2s in the membrane of the mitochondria, and you have to outnumber that by the pro-apoptotic BCL2s. So it's almost like a stoichiometric 1-to-1 thing where the anti-apoptotics will protect the cell until you've got more of these pro-apoptotics, and then cytochrome C gets released from the mitochondrion, and also a lot of calcium too. That's also an important part of this. Okay, so cytochrome C gets released. Then what happens? It binds to an adaptor protein called APAF1, and APAF1 then brings in procaspase 9, which is an initiator caspase. And these come together in a really interesting and complicated complex. It's not just three things coming together. It's like multiples of each, and they make this big wheel, the wheel of death. And this activates caspase 9. And caspase 9 then activates caspase 3. So we're back to caspase 3. Okay. So that's the BCL2 story. You've got your pro and your anti-apoptotic BCL2s. So how are these regulated? Pro-apoptotic BCL2s, you got to figure they're always there, otherwise your cell would die. I mean the anti-apoptotics. The pro-apoptotics, though, are going to come up because of a problem. We know PUMA comes up because of DNA damage, and some of these other ones are coming up for other reasons that we'll talk about. One is the withdrawal of trophic factors. So let's go back. Let's go way, way back to, what was it? Chapter 14. So remember cell crawling from chapter 14. I said that cell crawling is based on integrins coming together in focal adhesions. And those focal adhesions are going out and grabbing extracellular matrix, fibronectin and collagen. And they pull themselves along, they let go, they pull themselves along. So that's one function of focal adhesions. But I believe at the time I even said that these focal adhesions are also signal transduction complexes. So when that integrin goes out and grabs that extracellular matrix, it activates signal transduction inside the cell that tells it, look, we're attached to stuff, we're safe, everything's okay. And that is called a trophic factor. Okay, the trophic factor tells the cell that it's in a good condition and it can survive and it should be fine. Okay, that's one type of trophic factor. Another is growth factors. So if a growth factor comes, it will, activate signal transduction, and like what we saw with BAD, that signal transduction phosphorylates your pro-apoptotic BCL2s and turns them off. So as long as you're in a situation where you've got some kind of communication going on, you're going to be turning off those pro-apoptotic BCL2s. So let me see what I got for slides here. It looks like I see something about a slide. Okay, we'll come back to that. Okay, back it up. Back it up. Okay, so where the hell was I? Okay, so here's why that is. We'll talk about this again in the next chapter on cancer. If a cell is in a tissue, then it's probably okay. But if you got a cell like an epithelial cell that's no longer attached to a tissue, something's wrong. It should be attached. And cancer cells are known for letting go and not being attached to things. So this is like a backup plan. If those integrins are no longer attached to extracellular matrix, the cell dies. If the cell is no longer in communication with other cells through signal transduction, it dies. And that's kind of like another cancer protection. Another one of those things cancer cells have to get over in order to survive. So like most cells, to be metastatic, cells have to go into the bloodstream and go somewhere else and then set up a new thing. 99.999% of cells, when they go into the bloodstream, are no longer connected to anything and they go into apoptosis. So it's a protective mechanism. Okay, what the hell? All right, so let's go back to our slides. Okay, so this is that extrinsic pathway, and I've got a neat animation about that, but I don't want to break my stride, so I'm going to come back to this. We'll look at animations after I get through yakking. Okay, so absence of survival factors, which could be either no longer connected to extracellular matrix or no longer getting any growth factors to keep you alive. What's going to happen is those pro-apoptotic BCL2s will increase in the cell. Normally your BCL2 or your anti-apoptotic BCL2 is inhibiting it, but once it becomes this outcompetes that, you get release of cytochrome C. Cytochrome C then forms this wheel of death with APAF1 and procaspase 9. And once you get that, procaspase 9 activates caspase C3, and we're back to where we were before. So this is what I meant when I said they both converge on caspase 3. So either whether you're going on this pathway or this pathway, you're going to get caspase 3. So this is the extrinsic pathway, and now we've got two intrinsic pathways. Here again, it's intrinsic because this isn't another cell telling it to die. This is the cell realizing it should die. The other path to this is your P53 damage, or your DNA damage and P53 activation. That brings up PUMA, which also causes the intrinsic pathway to go on. So we've got two ways to get to the intrinsic pathway and one for the extrinsic pathway. And here's our apoptosome. Let's watch this video because this is, again, Drew Berry, and it's pretty crazy. Crazy. Hang on a sec, we'll get there. Video unavailable. What in the name of God is going on here? How dare you? All right, let's try this one.
One way.
All right, new share. This isn't the one I want to show you, so I'm going to look for that one.
The development of cancer is prevented is via the death of defective cells. A cell that becomes mutated or damaged will first attempt to repair the damage. If that is not possible, the cells will commit the cellular version of suicide. An orderly process called apoptosis leads to the breakdown of key cell parts and the death of the cell. Cancer cells lose this critical capability and will continue to divide. This can lead to the accumulation of cells that can become more and more abnormal.
And that's it. Okay. Escape. Let me try something. Oh my God. How does this person come up for apoptosis? Come on, YouTube. You can do better than that. What are all these people? Oh, Drew. Who is Drew? Oh, Drew Barrymore. Oh, Christ. Christ almighty. Let's just do apoptosis animation. Drew Barry. See? Aha. Aha. Here it is. This is the one. Watch the change in magnification. So this is kind of microscopic right now, and it goes submicroscopic eventually. Here's your disease cell over here. Here comes the killer T cell. Hello, my friend. Whenever someone calls you my friend, you know you're in trouble. Okay, so here's FAS. You'll see FAS ligand come in and bind to it. FAS ligand. Ooh. Now what? Let's go inside the cell. Ah, here comes FADD. FADD, fast associated death domain. So here's caspase 8 activating caspase 3. Mitochondrion, which is now releasing its cytochrome C. It's the wheel of death. It's just a beautiful structure. Comes caspase 9, pro caspase 9. I love the sound effects here too. This is a microfilament. Or it used to be. Now the cell is going, "Ooh, I don't feel so good." And there's your apoptosis. Cytoskeleton broken down, cell falling apart. All right, I have to stop. I can watch these all day. All right, let's see, what else do I got for you? Let's get this out of my way. All right, let's go back to this. I believe, oh, here we are. Oh. So yeah, that's all I have for this chapter. That's all there is. But then there's this figure, which I absolutely love. So people say, what do you think you are, a rocket scientist? Cell biology is so much more complicated. Look at this. This is your focal adhesion here. Talon, Paxillin, P130 CAS, vinculin. And then you're activating this guy here, which is FAK, focal adhesion kinase, and SARC, another kinase, which is activating PI3 kinase, which is activating RAC, as in RAC-RO and CDC42. So you know a lot of this stuff already, but it just looks complicated. Here's a receptor tyrosine kinase, GRAB2, SOS, RAS. Where's our RAF? They're around here somewhere too. Phospholipase C, diacylglycerol, protein kinase C, protein kinase A, activating another G protein. So a lot of these pathways you know. What you don't know is they all work together. It's just very complicated the way everything comes together. But anyway, that's what I have for you today. Any questions or comments? So 24 people left. Alexa gets 3 bonus points and someone else got 1.
Samantha.
Sammy. Okay, so that's all I have for you today. Yeah, we will meet back here again next Tuesday and we'll start talking about cancer and cancer cells. Okay? And then I guess the following week is our last week, so we'll have a review on Tuesday and then exam on Thursday and then final on finals week, which is the following week. Okay? Looks like we're going to end on time. All right, guys, have a great weekend.